Alrighty, everyone. I am going to be recording here the first online lecture. Today we're going to be covering the end of Chapter 10, which is Evidence for Evolution. So we have these five that we're going to be talking about today. These are the primary ways that scientists have found supportive evidence for evolution. So we have the fossil record, biogeography, comparative anatomy and embryology, we have molecular biology, and we have laboratory and field studies. Sometimes when an organism dies, it is trapped in loose sediment. If scavengers do not disrupt the remains, they may fossilize. So we have within the sediment, the soft tissues can sometimes be replaced with rocks, and the harder tissues are slowly replaced with rock within the sediment. So a fossil is now completely made of rock. But what we can see is some of the patterns of sometimes soft structures like feathers, shells, and bones are very easily preserved. So it's important to remember that as we look at fossil records, that only structures that can leave fossils will leave fossils. So we unfortunately don't have a fossil record for a lot of organisms like jellyfish, other soft-bodied organisms that don't leave behind uh, hard physical remains are very difficult to find preserved specimens of in the fossil record. Um, it's also important to remember that we need very specific conditions for this to occur. Generally, we need loose sediment. A lot of times this is going to be around bodies of water, so river systems, oceans, lake beds, that kind of thing, where the sediment is soft enough to sort of trap the body of an organism without having scavengers come and disrupt the body. Um, what it also gives us information about which kind of organisms lived in which regions as well. That can give us an idea of what the ecological conditions were at the time too, so we can guess about the nature of other organisms based on features of an organism. We can also guess about what kind of environments were there based on whether or not there was wings, fins, or other features present. So one of the ways we can date fossils is using a process called radiometric dating. So all isotopes are elements with the same amount of electrons and protons, but different amounts of neutrons in the nucleus. So they'll have the same atomic number, a lot of the same electrochemical properties, but they will have different weight. Uh, the neutrons will give weight but won't affect other things related to the atom. So what we can do then is detect some of these, the frequency of these isotopes, these atoms with different numbers of neutrons with different weights, because they decay at a very predictable rate. Based on the rate of their decay, we can predict the age of a given fossil or a given sediment layer. So these fossils will generally gather up in a layer that we unearth. So we can date a lot of that because of the rate of decay of these atoms. All right. So we just gave a whole bunch of fossil information. Now we're moving on to the second topic, which is biogeography. Biogeography is the study of the distribution patterns of living organisms around the world. So one instance of this we can see in figure 1036, which is honey creepers in Hawaii, which adapted to new environments, but still have a lot of shared characteristics with what we believe to be the ancestors of these organisms. So one group of finch colonized one island, then they kept moving from island to island. So they're geographically isolated via distance, but so they were able to get all these different forms, but they're still able to spread to those islands initially. All right. So we also have comparative anatomy. So a lot of components differentiate at the embryonic level. So most embryos have a lot of similar features, and then as the organism matures, those more and more differences appear. But we do see a lot of structures that are analogous or homologous between these organisms. So the same derived piece that then becomes differentiated later. For example, human embryos still have gill pouches, and that can be traced back to our ancestry to early amphibians. The origin of tetrapods, four-limbed organisms. We share a lot of characteristics with four-limbed organisms. All right. Homologous bone structures is another comparative anatomy tool. So the bone structure to a lot of, specifically with tetrapods, our four limbs can be compared to another four-limbed organism. So humans have the same setup for arm bones as front leg bones for a horse, as the wing bones for a bat, and actually crunched down and then sort of stretched out later, the fins of a porpoise. The event where so four-limbed organisms colonized the shores of the early Earth, but later, much later on, a group broke off and became aquatic again, and those are the cetaceans. Those are dolphins, whales, narwhals, other seal. I'm actually not sure on seals, but either way, dolphins and whales certainly fall under cetaceans. But anyways, with that, let's look at a example. So we had features that looked similar and have a shared background. Now we're looking at an example of something where it is a feature that shares a function, but they have different evolutionary backgrounds. They evolved separately from one another. So convergent evolution is an example of a characteristic or feature that is similar in function but has a completely separate point of origin. So in the case of uh, insects, they developed flight long after they separated from what would eventually become mammals. Uh, Vice versa is true as well. From uh, Mammals developed flight completely separately from insects. So one of the things, actually, sorry, we see with convergent evolution oftentimes is we see a trait that confers a large amount of fitness. So the ability to fly is opened up a lot of possibilities for these organisms. That's why we see flight occurring many times. We see the evolution of eyes, cells that can detect light. We see that occurring many times in evolutionary history as well. All right, so molecular biology is the study of DNA. So specifically, when we're talking about molecular biology, we're talking about generally speaking of the genetic code of an organism. That's what they're studying, or what that code does. So more DNA codes are shared between organisms which are more closely related, meaning they diverged more recently, speciated away from each other more recently. So as this graph is showing on the left, we see that 146, let's see, when we're looking at, so we're specifically looking at the structure of hemoglobin. So if we compare human hemoglobin to human hemoglobin, we see all 146 match as they should. When we look at rhesus monkeys, hemoglobin, by the way, is the protein that carries oxygen in the blood. So hemoglobin is very similar in rhesus monkeys. Then we go to dogs, it's a little more different. Birds, a little bit more different. And then lamprey, more different still. So we can even get examples of convergent evolution happening at the molecular level when we're looking at proteins. Sometimes proteins with similar functions have completely different origins as well. Sometimes they can just stray so far apart from each other that they're virtually irrecognizable from one another as well. All right. So we've talked about, let's go back to the beginning. We've talked about our fossil record. We've talked about biogeography. We've talked about comparative and anatomy and embryology. We have talked about molecular biology. Let's talk about some field studies. So some examples of field studies are antibiotic resistance. So we can track evolution as it occurs within our society. So antibiotic resistance has evolved in Staphylococcus aureus, a strain of bacteria that causes illness in humans. So when you put on a little disc of antibiotics, that'll sort of leach out into that agar, the gel it's sitting in, and it'll create a zone of inhibition, a kill zone, that the bacteria can't grow on. However, if you take an antibiotic-resistant bacteria strain, it is completely resistant to the penicillin. It doesn't even, the concentrations have to be incredibly high in order to kill it, to the point where it wouldn't be useful to us as a medicine. Oftentimes it's complete, it can be even complete immunity to the antibiotic. So yeah, we have an example where we put a selective pressure on these organisms, and they responded through brute force and lots of chance, eventually developing resistance to our drugs. So we're constantly modifying penicillin in order to get new versions of penicillin that work against bacteria. That is the current race of antibiotics. Actually, antibiotic resistance and antibiotics in general is one of the things that CRISPR is being looked at as like a possible tool for treatment of antibiotics, or of bacterial infections. Looking at creating custom antibiotics, essentially. All right. So antibiotic resistance is one way which organisms can differ. Another possible way is with, we can see just sort of the change in characteristics. So even if we're not having a speciation event here, we can see the change in characteristics between different types of, or the same species of grass, the same exact seed being placed on the same field, but it resulting in different characteristics when we treat the grass differently. So the example is in a golf course, we have the rough, the fairway, and the putting green. And because the putting green is cut so often, it has a much higher or a much quicker reproduction rate than the rough. The putting green is getting put through the selective pressure where only the grass plants that produce seeds the quickest will be the ones that create seeds and go on to the next generation, whereas that's not a concern in the rough. But that pressure that's been put on the putting green, and to a lesser extent the fairway, has caused a rapid increase in its life cycle. The putting green grass goes through its life cycle much quicker. So with that, so we have all these examples of some traits that are examples of convergent evolution. So getting away from our ideas of sort of evolution in motion, let's look at sort of the history of evolution. So similar to our wings on our bats and dragonflies, think of a trait that is very important to one organism that is similar in another organism maybe, and determine whether or not it is a homologous structure, so whether or not it came from the same evolutionary landmark, same shared evolutionary history, or whether it evolved separately, so whether or not it's a convergent structure. So convergent evolution or a homologous structure is what we're going for here. Did it evolve separately, or did it evolve at the same time and then they speciated later? I want you guys to give me an example for the animals. I want what you found out about their backgrounds, and just make sure you cite those sources as well. The post for this will be on the discussion forum. As always, if you have any more questions, please feel free to contact me, and have a great day. Stay safe, everyone. Bye.